Hi students, let's talk about regulation of gene expression. So for this, we got to remember what we've been learning so far in this unit, the central dogma of biology. So this is of course that DNA is the directions to make RNA, and that is transcription. And then RNA is the directions to make proteins, and that's translation. And the reason why proteins are so important is because the proteins that a cell has are going to determine the traits of that cell. In other words, its physical appearance, its structure, its function, all that good stuff, right? So we should know by now that genes are sections of DNA that code for specific proteins. So just to remind yourself, there's a nice little picture of a cell. Inside that cell, if it's a eukaryotic cell, is a nucleus. And inside that nucleus are chromosomes, which are just big pieces of DNA. So this DNA has a bunch of segments on it, which are genes, and each gene can code for a different type of protein. So in this example, we have like this first gene here, which is coding for this green protein. This middle gene codes for this red protein, etc. So that's what I mean when I say genes are sections of DNA that code for a specific protein. But what I want you to think about today is, to start, where did you get your genes from? Yeah, so every organism inherits their genes from their biological parents. So in other words, we have an egg cell and a sperm cell, and during fertilization, those two cells come together and make a zygote. So that's what we can see down here. The sperm is going to come and fertilize this egg, and it makes sort of like one complete cell. And then that cell is going to divide and divide again and divide again. And over a whole bunch of cell divisions, that will eventually become a little baby person. So in other words, every single cell in your body came from the same starting cell. It all started as one egg and one sperm that came together to form that zygote, which eventually became you. So we had this one zygote, it divided, made some more cells, those cells divided a lot more, and eventually you had an organism, a chick. So keeping that in mind, I want you guys to make a prediction. What do you think? Do you think the DNA in all of your cells is different or the same? Yeah, so it turns out that all of your cells actually have the same DNA. And that's because they came from the same starting cell. That starting cell was a mix of biological or DNA from your biological mom and DNA from biological dad. And that's all the DNA that cell ever had. And that's true for all the cells that came from that cell. So all the cells in your body actually have the same DNA, but they are drastically different. So for example, here's a picture of a nerve cell, and look at how those nerve cells are really long and spindly. That's because the function of nerve cells is to communicate over long distances. So they have a very specific structure that makes them long and spindly to help them with that function. If we compare that to a muscle cells almost look like accordions so that they can expand and contract because that's what our muscles do, right? So there's completely different structures and functions between these two cells, but they have the same genome. They have the same DNA. So how the heck is that possible? What do you think? So turns out it's possible because even though each cell has the same genome, only a small number of those genes are expressed, meaning that only certain genes are turned on. So for example, in this picture, imagine we're comparing now a liver cell compared to a neuron. The liver cell is going to have all the same genes as the neuron, but in this liver cell, it's only going to make genes that livers need. So for example, alcohol dehydrogenase, that's an enzyme that's important for breaking down alcohol. That's one of the things your liver does. So in your liver, that gene's going to be on. On the other hand, this neuron-specific gene called a neurotransmitter, that's not going to be turned on in the liver because the liver doesn't need that. And vice versa in the neuron. It once again has both of those genes, but the neuron is only going to have the neurotransmitter gene turned on because that is a gene that's important for its structure and function. So in other words, gene expression, meaning which genes are turned on and making proteins, gene expression is going to determine which proteins a cell is producing, and the type and amount of proteins that a cell makes is what determines the traits of that cell. So key points here. All the cells in your body have the same DNA, but each cell only has a fraction of all of those genes turned on. A gene that is, quote, on is going to be transcribed to produce mRNA, and then that mRNA is translated to make proteins, right? So a gene that is on, we say it's expressed, meaning that gene is actually being used to make proteins. And whatever proteins are being made, well, that's what gives a cell its unique traits. Okay, so we should be familiar with this picture here, right? So DNA, RNA, protein, we already know this. So it shouldn't come as a surprise that how we regulate gene expression can happen in two ways. We can either regulate how much transcription is happening, in other words, how much RNA we're making, or we can regulate translation, in other words, how much protein we're making. So transcriptional regulation, which is just controlling how much DNA to RNA. So like really we're like either making lots of mRNA or not making it. Really it all comes down to RNA polymerase. RNA polymerase, that's the enzyme that is actually making RNA. It's the one that actually copies the DNA. So the way that we regulate transcription is essentially just by making it easier or harder for RNA polymerase to access the DNA and actually transcribe those genes. So one way that can happen is to understand that you kind of have to remember what it looks like inside of a cell. So here's a cell, and here's our nucleus inside the cell. And there's a bunch of chromosomes in there. Chromosomes are just made of DNA, right? So here's our DNA. And in your nucleus, the DNA is actually wrapped around these special proteins called histones, sort of like a spool of thread. And the DNA can either be wrapped really, really tight, like in this picture, or it can be looser. So see over here where the DNA is more loose, there's a lot of like unwrapped DNA here. This DNA is accessible to RNA polymerase, so those genes can be turned on. But here where the genes are like really tightly wrapped, RNA polymerase can't get to that DNA because it's like all clumped up in those spools of thread. So those genes are going to be turned off. They're not going to be expressed just because we can't do any transcription. So that's one example of transcriptional regulation. And then, of course, we can also have translational regulation. So this would be regulating how much mRNA is used in order to make proteins. For this, you have to remember that ribosomes are what are translating mRNA in order to synthesize proteins. Usually, the way translation is regulated is by destroying mRNA. So if you have less mRNA, you just can't make any protein, right? And we've already just barely learned a little bit about this. If you did the reading assignment on Friday in class, then you learned about RNA processing. So this is just we take RNA that's made during transcription, and then that RNA goes through a series of steps such as splicing. This little bit's added on the 5' end called a cap, and a poly A tail is added on the 3' end. And the whole point of these two things, the 5' cap and the 3' tail, is just to make the mRNA more stable, meaning it's going to last longer before it breaks down. And that's a type of translational regulation. So in summary, all the cells in your body have the same genome, meaning the same set of genes, but each cell is only going to express or turn on certain genes. Gene expression can be regulated at the transcriptional or the translational level, meaning that in order to figure out if a gene is turned on or off, we can do that either by regulating how much transcription is happening of that gene or by how much translation is occurring of the RNA made from that gene. And there we go. There's a little bit of an introduction to gene expression.